Dr. Pritt, welcome to the Tick Bootcamp podcast.
Thank you. It's a pleasure to be here.
We're really excited to have you. We've been geeking out on you and all of your accomplishments for quite some time now, so it's really a blessing to have you with us. Why don't you first share with us, or I should share with our listeners, where you're calling in from?
Yeah, I'd love to. So I'm at the Mayo Clinic in Rochester, Minnesota. And at the Mayo, I am the director of the clinical parasitology lab, and I also co-direct our vector-borne disease testing services. So anything that is transmitted by any type of vector, including ticks, mosquitoes, and other arthropods.
Okay, so we're gonna focus first on ticks. We may talk a little bit about mosquitoes because there is some
Okay.
controversy that we've stepped into recently. So we really certainly like to get your opinion on one thing as it relates to mosquitoes. But let's say focused on you first. Can you share with our listeners what your educational background is?
Yeah, sure. Well, I am a physician by training. I went to medical school at the University of Vermont, a beautiful state for those of you that have been to Vermont. Unfortunately, seen a lot of tick-borne diseases as we are throughout a lot of the United States. And after medical school, I decided to specialize in pathology. So, pathology is the study of disease, it's very broad. I decided to focus more on infectious disease and so decided to do a residency in pathology and then a fellowship in infectious or in clinical microbiology. Now, after clinical microbiology, you talked about geeking out. Well, I geeked out over parasites, ectoparasites. I went to the London School of Hygiene and Tropical Medicine and got a master's degree in parasitology with a healthy dose of entomology. So that's my training.
Holy cow. I don't know that someone could have a more, I guess, diverse set of training and educational experiences to bring them to the Lyme community. You really do have sort of everything sort of touched upon. Matt and I really love pathologists. One of our mentors, unfortunately, he's passed away, is Dr. Alan McDonald. Dr. McDonald, of course, was a really cool medical detective just like you. Talk to us a little bit about what it's like to be a medical detective, specializing in pathology.
Yeah, sure. So pathology, as I said, it's a really broad field, but I decided to focus on the pathology of infectious diseases. And we're the doctors that you don't always see. We're sometimes behind the scenes, but we're responsible for all of the tests that people get. When you go to your doctor and you get your blood drawn, that's going to a laboratory that probably has a pathologist overseeing its testing or another doctoral level individual with specialized training. So I am responsible as the medical director for overseeing all the tests that happen in my laboratory. I might not be the one on the benches doing the tests. I have a really awesome group of talented technologists, but I'm ultimately responsible for the quality of every single result that we release. And every single one of those results, they're patients. They're all my patients. So every barcode is a life, is what we say. Every single one of them I'm touching. And as a pathologist, I know that the information that we're generating in the laboratory makes up the majority by far of the information in a patient's medical record. It's about 70% of all the information in a patient's medical record is laboratory driven, is coming from the laboratory.
Wow, cool. So let's talk about some of your accomplishments. When Matt and I were doing our research, we came to understand that you have actually discovered new strains of various types of tick-borne illnesses.
Why are you
Why
stuttering,
don't
Rich?
you?
You can't say the names. Is that why you're stuttering, Rich?
I
Can you
cannot.
come on? Try to pronounce them, please, because I can't.
I
Dr.
I can
Pritt's gonna
say
have to
them.
say them for us.
All right.
I can say them. It's no problem, Rich. Yeah, well, part of it was being in the right place at the right time. And we're in Minnesota, the Mayo Clinic in Rochester, and there's a lot of tick-borne diseases in the area. We are also an international reference laboratory. So we get specimens sent in from all over. But it turned out these new tick-borne pathogens we detected They're upper Midwest. They're right at home here. So it turned out the first one that we discovered was from Eau Claire, Wisconsin, which is a fairly large city in Wisconsin. And we have a Mayo Clinic branch there. And some of the tests that we had developed here in Rochester were being performed there for a number of different pathogens that we associate with ticks. Lyme disease, would be one disease caused by Borrelia burgdorferi. We are testing for Anaplasma phagocytophilum and Babesia. Well, it was two astute technologists that were on the bench and they noted an atypical result. So shows you the importance of boots on the ground, the people that are right there doing the testing. They could have ignored it. They could have just said, this is nothing. Let's just report it out as negative. But they said, no, this is something unusual. And they raised the flag and of alarm and then contacted me. I looked at it, we repeated the testing. Sure enough, we kept getting this atypical result. So I reached out to our state and public health labs, the CDC. We did a lot of additional testing. And sure enough, it was a novel pathogen called Ehrlichia murus occlurensis. Now, people that know of Ehrlichia, they probably think Ehrlichia chapiensis, Ehrlichia ewingii, but Ehrlichia murus, murus comes from it's mouse, the Latin root. So it's a mouse organism that usually found in mice, and it had never been found in North America. It was only found from Russia and Asia. So we're like, what is this doing here? Well, it turns out we just didn't have the tools to detect it. So it also shows the importance of these novel pathogens. There are probably more out there that we don't know exist yet because we just don't have the tools or we're not testing the right people. So that one was really fascinating. We continue to see cases of this every year, not a lot, but we detect it. So we ended up calling it Ehrlichia murus occlurensis. And we called it occlurensis after Eau Claire, Wisconsin, because that's where it was first detected. So that was organism number one, Rich, but I don't know if you want me to just keep going.
Please
Real
do
quick.
keep going. I have
right.
questions about all of them generally, but I'd like you to first outline
Yeah.
for us all of the new microbes you've discovered, and
Okay.
then we'll talk about how you discover them and how you discover a new one. And I have questions about what else is probably out there.
Yeah, sure. I'd be happy to keep going. So I won't go into all the nitty gritty detail unless you want them, but it was only a few years after we published our findings and we had sent out public health alerts to everyone saying, hey, there is a form of ehrlichiosis in the upper Midwest, and you probably didn't know it was here. So be aware, your patients may have it. And it was only a few years after all of that publicity, and we made sure everyone, we could get the word out. We published our paper. And then all of a sudden, we detected another atypical result, but this time it was on a test for Borrelia burgdorferi, which most of you probably know is the main cause of Lyme disease in the United States. Well, this was not Borrelia burgdorferi, and it was not Borrelia guerini or Evzelii, which caused Lyme disease in Europe, but it was something novel. So we kind of did the whole thing. At first, I thought, oh, wow, again, really? But I had all the contacts. I knew the people at the public health labs, the universities, the CDC, my staff, my researchers, my developers were really talented. So we knew how to go about this, and we dove right into it and did a lot of additional testing. Sure enough, another novel organism. This one we called Borrelia mayonii, and we named it after the Mayo brothers. So Mayo and mayonii is how you say it. This is the second proven cause of Lyme disease, well recognized in the United States. Also in the upper Midwest, although both of these organisms now, Ehrlichia murus occlurensis and Borrelia mayonii, we're starting to see a smattering of cases in New England. So it probably has a wider distribution. Now, Rich, you had asked other organisms that we've detected. There's a handful of them. I've been partnering with the CDC now, and we've done some great work together. We've detected an Anaplasma bovis-like organism. Doesn't cause Lyme disease, but causes anaplasmosis in human blood. We detected Borrelia portoricensis like organism, as the name would imply, portoricensis, it's Puerto Rico. So this is an organism that had only been detected down in Puerto Rico, and now all of a sudden we're detecting it in a patient specimen in blood. And then we detected Borrelia johnsonii, never had been detected in humans before, except up in Canada, it was detected in ticks. So these are things that some people had found before in ticks, but never in humans. Now we're finding them in humans. We have a novel rickettsial organism. It does make you wonder, doesn't it? What else is out there?
Well, it makes me wonder many things. So let's first talk about geography. So you shared with us that you were discovering different microbes in different geographic areas.
Mhm.
Do you think there's a reason why some microbes are isolated to certain geographic areas, or are you just detecting them in those geographic areas and you expect, like just like any other microbe, they're probably going to migrate, however they migrate, on the backs of
Yeah.
deer, birds, however they might migrate?
Well, initially we thought that both of our first two organisms we detected, we thought they were only in the upper Midwest, Minnesota, Wisconsin, maybe North Dakota. But as I mentioned, we're detecting them now in New England, but not in very any large number. Most of them are in the upper Midwest. So why would that be? Why would we have that geographic restriction? It might have just been that we have some subtle differences in the upper Midwest. For example, we get really cold winters and really hot summers, and that changes the way the ticks feed on their different life cycle stages. So we have what we call co-feeding. There's more larvae and nymphs co-feeding at the same time. And so they can easily pass organisms between the two different life stages. That may play a role. Um, it could also be that these organisms, these new ones, they're just not present in very high numbers, thankfully. So it's kind of hit or miss whether people actually get infected with them. And then, of course, a lot of people don't get a diagnosis, they don't get a blood drawn. Um, it doesn't go to a laboratory that is capable of detecting the organism. So that's a lot of just chance. And some patients just get a dose of antibiotics, makes them feel better, probably kills the organism, and then they never even knew they had this novel thing.
Okay, so let's focus on the co-feeding first. What
Yep.
is co-feeding and why is that significant in the transmission of these microbes?
Okay, so the way it works, co-feeding is when you have different stages of ticks feeding on the same animal at the same time. So ticks start off as eggs and then they hatch into larvae, and then the larvae take a blood meal. And yes, that's exactly what they call it. So it sounds gross, but yeah, they take that blood and that's their meal. And then the larvae can then shed their skin and molt into a nymphal stage. And they go from having six eggs legs to eight legs. And then that nymph takes another blood meal and then it can molt into an adult. So every stage needs a blood meal. That means that some poor mammal, rodent, human, something is going to get bitten so they can get that blood. Now, let's say that a larval stage bites a mouse and the mouse has one of these novel pathogens like Borrelia mayonii. It picks it up, but then it has to bite another host in order to be able to transmit it. So it means that it has to bite the mouse. It has to, in order to pick up the organism, it has to have bitten the mouse during the stage where the mouse actually had this organism. Well, what we think is maybe some of these organisms don't circulate in blood for very long and then get cleared by the mouse. But if you have co-feeding, let's say you have a nymph that is infected, it bites a mouse, it injects Borrelia mayonii into the mouse, and there just happens to be a little larva right next to the nymph that is feeding at the same time. It picks up the Borrelia mayonii. It doesn't matter that maybe the mouse clears it right away. By feeding at the same time together, they basically can transmit the organism to one another. That may allow the organism to survive in a place like the upper Midwest because of that co-feeding. And we don't see that same overlap with co-feeding in the Northeast. Now, this is all a hypothesis. And I should note that we actually do see some of this organism now popping up in patients in New England. So it could just be a faulty hypothesis, but we do know there are differences between the way ticks feed in New England and the upper Midwest.
Help me with that. I don't understand the difference between why ticks would feed differently in the upper Midwest versus New England versus Long Island, where Matt and I are.
Okay, well, first of all, I should say I'm not an ecologist. So all the ecologists out there are probably listening and saying, oh, she got that all wrong. But it's my understanding with our really cold winters and our really hot summers, ticks don't like they can't survive freezing, and they don't like it when it gets too hot and dry either. So that means that there's a narrower window where they can actually be out and about and not dry up and die or be frozen. So you get a narrower window, and during that same window, the larvae, the adults, and the nymphs all have to try to feed at the same time. So you're probably going to get more co-feeding because of that narrower window. Now,
Okay.
that will change over time if with global warming, if our winters get warmer, then maybe you have a longer season and spring comes earlier. But historically, that's the way it's been.
Okay. So now, um, do you suspect that you were finding something that hasn't been found somewhere else, not only because of the co-feeding issue that you defined for us, but because of the testing issue? Meaning, you for the first time, or your lab for the first time, observed these pathogens, and obviously that may be these pathogens may have been in other places and they just weren't observed, and there certainly wasn't any testing to observe it. So do you think part of the reason why you may have only been observing it where you are is because you're the only ones that saw it, and because you're the only ones that saw it or knew to see it, that it really is in many other places, it just hasn't been observed yet?
Well, so yes and no. So first of all, you're correct. You need the right test. And part of it is just having the test in the right time in the right place, and you're testing the right people and you pick it up. And we happen to have a test that is broad enough to pick up novel organisms. And I'll tell you, that's the direction the field's going in. Broad tests that can detect anything. So we're probably going to see more things getting picked up with our broad tests. But back in the early 2000s, when we detected the first organism, Ehrlichia myricoclorensis, we were probably the only lab out there that had this type of test. But then, Rich, to clarify, as a reference laboratory, we do get specimens from New England. So even though we test a lot of patients from Minnesota and Wisconsin, we test a lot of patient specimens from Vermont, New Hampshire, Connecticut. So we originally had over 100,000 patients that we had tested from New England, and we did not detect a single positive for these new organisms in that population. We
Interesting.
only saw it from patients in Minnesota and Wisconsin. But now, now we've been doing testing for 15 plus years, and every once in a while we get a rare positive. And sure enough, it looks like it is in New England, but perhaps just both organisms, probably at very low levels.
All right, so let's focus on species of ticks. So
Mhm.
obviously, different species of ticks are going to harbor different microbes.
Yes.
Now, do you think that's also an issue why perhaps you have the pathogens that you're observing where you're observing them and not observing them in other places because there are different species of ticks that are native or just populating different geographic areas?
Well, it is a factor. It's true. There are some ticks we just don't find in certain places. But you heard me talking a lot about New England and the upper Midwest. And we both have the so-called deer tick, black-legged tick, Ixodes scapularis. That's present in both places. So you can't say that that's unique in one place or the other. Um, it it is true though that the Lone Star tick, which you've probably heard of, Amblyomma americanum, that is much more prevalent now on the East Coast, going right up into New England. And we aren't seeing it yet in Minnesota, at least not in any large numbers. We don't think the population is actually established, even though we occasionally detect it. And it's probably not that far up into Wisconsin. But these two organisms I told you about, Borrelia mayonii and Ehrlichia murisochlorensis, every test we did on ticks only detected it in the black-legged tick. We never detected it in the Lone Star tick.
Okay.
So you
yeah.
can't really blame it on the ticks. The ticks are the same between the two regions.
Okay. Well, at least the black-legged tick.
At least the black-legged tick. Right.
Okay, so let's now talk about the black-legged tick generally, and the number of microbes that the tick can harbor. Matt and I, on a couple of occasions, have talked about a study we came across a couple of years ago, and I don't remember who the researcher was, so I apologize, Mr. or Ms. Researcher, whoever you are that that did this work. But we came across a study that indicated that a black-legged tick could harbor as many as 200 different microbes. Are you aware of that study, and is that consistent with what you're finding?
Well, so it's a good question. I'm not familiar with the study, but I'm not surprised. But one thing to keep in mind is just because it harbors a microbe doesn't mean it could transmit it or that it would be a pathogen. Because think of all the bacteria that live in our gut. We harbor those, but we don't pass them on to other people usually. Or at least, we're not biting people usually and spreading
Right.
germs that way.
I mean, that's what I was thinking. We're generally
Yeah.
not biting people and spitting in them, but
Yeah, exactly.
and of course, and remaining attached for 3 or
Right.
4 days. Right.
So let's start with the pathogens we know, though. So I'll just do a count on my fingers. We have in the black-legged tick, we know there's Borrelia burgdorferi. That is a pathogen that can get transmitted to humans. Then we also have Anaplasma phagocytophilum, and we have Babesia microti. Now, those are the 3 big black-legged tick pathogens. But now we know that Borrelia mayonii is another one, and Ehrlichia murisochlorensis. So we've now added it up to 5 bacteria. We have 4 bacteria and a parasite. But then we also know Borrelia miyamotoi is in the black-legged tick. So there's another pathogen, another bacterium. We have Powassan virus, and that's found in the black-legged tick. So you can see how the numbers are starting to go up. We're mostly bacteria with a virus, Powassan virus. Now, are there 150 other organisms that you could find inside of ticks? Yeah, they're probably natural commensal organisms. Maybe there's some pathogens in there, or maybe they're just all part of the normal flora that live in the gut, the salivary glands of the tick. So those, it's hard to know what to make of. But there are a lot of people who do studies and look at all the different things you can find inside a tick. And you do have to clean the tick on the outside if you're going to test the tick, because they're dirty little critters. They, walk through the understory. They're covered in fungi and bacteria. So you also have to consider just what their external normal flora is as well.
Okay. So, do you think it's possible that there are many other microbes that are being spit into us when we're being bitten by a tick? Specifically, we'll talk about the black-legged tick, since that's what we're focusing on now. That we just haven't discovered and could be pathogens that are causing us to suffer illnesses.
I do think so. Yes. There are probably some that are in small numbers, not very prevalent, like Borrelia mayonii. It's certainly not the major cause of Lyme disease, even though a small number of people do get infected. And so then we're probably just not picking it up, or people get treated with doxycycline, they get better, and they never know that they had a novel pathogen that no one knew about. So,
Or could another possibility be that they're getting sick, it's not diagnosable, it's not being treated, and it is one of the reasons why they continue to have a long-term chronic illness.
yeah, it could be. It depends on the type of organism. Doxycycline is a good antibiotic, and it will cover a lot of your tick-borne bacterial pathogens. But if it's a virus, it's not going to do anything for that. If it's a parasite, not going to do anything for that. So you have to have the right drug on board as well.
And doesn't the timing of taking the drug going to be
Yeah.
significant as well, right? I mean, yes, one of the wonderful things of about doxy, and there are some things that are not so wonderful, but let's just talk about the wonderful characteristics, is that it's a broad spectrum antibiotic, and because it's a broad
Hmm.
spectrum antibiotic, if it's taken soon after a tick bite, it's generally going to be an effective tool to
Yeah.
kill a lot of bugs that have been spit into you. But if you suffer a tick bite and you don't find the tick, if you
Right.
suffer a tick bite and you don't discover the illness until some window of time after it's been able to spread through your body, then doxycycline is likely not going to be an effective tool in killing this broad spectrum of bugs that could have been spit into you.
Yeah, I'll mostly agree. And not a treating physician in that I'm not prescribing doxycycline. But I will add the caveat that even if it kills the bacterium, if it's given later, it might have already done the damage. The bacterium, if it got into your joint, okay, so Lyme arthritis, terrible condition, and you can treat it. And if you've already suffered a lot of inflammation, it may not go away for years. And you may have a swollen knee, and you can do PCR testing and still detect the DNA of the bacterium. There's some thought that the knee is a protected space. So even though the organism might be dead, it remains there. And there's been some new research showing that parts of the bacterium, their peptidoglycans, remain there and continue to cause damage because your body reacts to it and you get all this inflammation. So some of this is still under investigation and we don't know exactly what's going on. But bottom line, if the bacteria has been in your body and it started doing damage, you're going to have to heal from that damage. And maybe you kill the bacteria with an antibiotic, but your body's still going to have to heal. And then that could take a really long time.
Okay. So now you said that your lab was using testing that allows you to discover novel microbes. Tell us about that testing and why that's unique to your lab or to the labs that you work with.
Yeah, well, I'll start with the test that we used way back in the day, early 2000s. Well, it was actually sort of like 2008 or so, so late 2008, 9, where we first detected Borrelia mayonnae and Ehrlichia occlurensis. We were using tests called PCR, which I'm sure a lot of people heard of. Now, after COVID, a lot of people know what PCR is. It
Yep.
detects the DNA of organisms. And usually if you're detecting the DNA, that means that it was really there. It's not just detecting the host response, which can be misleading sometimes. So we detected the DNA. The way we had designed the PCR, we made it so that it could detect multiple closely related organisms. So let's take Borrelia mayonnae. We created this test that could detect Borrelia burgdorferi, Borrelia afzelii, and Borrelia guerini. That makes it a broad spectrum test, so to speak, or it's a multiplex test. It can detect all three. Well, because of the way we designed it, and we use something called melting curve analysis, we would expect peaks in certain temperature areas. It's complex to get into, but I'll basically just say that Borrelia burgdorferi, we would expect it to have a melting temperature peak of about 70 degrees. But what we actually saw was something that was a little cooler, and that was a unique result. So it's a type of PCR test that we were using. It was developed at Mayo Clinic, so no one else would have had it. It's a laboratory developed test, and we were finding an atypical result. So that was novel. That was something that other labs at that time, no one else had a test that would have detected this novel organism.
Okay, so I'm going to ask you to bookmark the testing for a second. Matt's going to want to join us in that part of the conversation, and I'm going to come back to the ticks for a little bit longer. Um, so you had raised the lone star tick, which is a very aggressive tick, and the
Yeah.
tick that we're seeing more of here on Long Island than anywhere else. The last three tick bites that I suffered here over the last couple of years were all lone star ticks,
Ugh.
um, and
Yeah.
and they're very very different. I mean, having grown up on Long Island, having been bitten by ticks many times in my life, and having picked ticks off not only myself but also of our dogs during the course of my life. I've witnessed very different behavior from the different tick different ticks. And what I find with the Lone Star tick is that it's a much more aggressive tick. And one of the things I put up on Instagram a couple of years ago, Matt will remember, is I found a Lone Star tick actually biting me while I was actually talking with Matt. We were actually on Zoom, and I had pulled the
Oh,
tick off. And then what I did was I went into my bathroom and I put the tick on the front of the sink and I turned on the camera and the tick kept running at me.
running
And when I
at you,
push it back
yes.
again, it was literally coming after me. And I pushed it back again and it came after me again and it pushed it like it was a very aggressive tick. And Matt posted it on Instagram and everybody was creeped out by, the aggressive
Yes.
nature of this tick. So can you talk about the nature of the different tick species, and do you think that's driven by the pathogens that they're harboring, or do you think the either aggressive or more passive questing behavior is a function of the species of the tick itself?
Yeah. Well, lots to talk about there. First of all, let me tell you that you're exactly right. Everyone is the people that study ticks have shown that these are truly more aggressive ticks, the Lone Star ticks. Some people call them the lions of the Serengeti Plains. They're the predators. They will track you down. They're
Yeah,
attracted to your CO2 that you release when you breathe. They're attracted to your heat, and they will actually they will chase you to some extent. They will go
yeah,
towards you
yeah,
as you identified. Whereas the black-legged tick does the questing behavior where it climbs up on a blade of grass and just hangs out with its little legs extended and it waits for you to come by. But these guys don't wait for you to come by, they'll go seek you out themselves. So, very aggressive ticks. Now, you asked, what are the differences between these different ticks? I will say it's not just that they're different species, they're a different genus altogether. So, they're all ticks. They're all in the Arachnidae family. They are eight-legged as adults, but they're very different, just like different spiders. You might have like a black widow versus some harmless gray spider, two completely different genera. So a lot of it just has to do with their genetic differences. And so because of those differences, they may be more adapted to being a vector for certain types of pathogens. That's why there's not 100% overlap. We'll see that the Lone Star tick can transmit Ehrlichia chapiensis and Ehrlichia ewingii, but the black-legged tick does not. So they're being different animals, really. They have different abilities. The organisms can survive in them and they can be transmitted, and not all ticks can transmit the same thing as a result.
Okay, so let me ask you to now focus on the different microbes that different ticks are harboring, and do you believe the microbes have an impact on the behavior of the tick?
Well, I can't say that the microbes themselves necessarily have an impact on the behavior of the tick, but it is true, being different ticks themselves, some ticks can harbor different organisms than others. So the Lone Star tick, of course, it's famous as the vector for Ehrlichia chapiensis, Ehrlichia ewingii, Francisella tularensis. Then there's the big one. It's associated with meat allergy. Something in its saliva when it bites you can cause you to form antibodies against alpha gal, which is a sugar moiety. That's a big one. And it seems to be the primary tick associated with alpha gal syndrome, meat allergy, so to speak. And then also the Lone Star tick has been associated with Heartland virus and Bourbon virus, whereas the black-legged tick has not.
Okay, so one of the studies we came across in the recesses of our memory indicates that black-legged ticks that harbor Borrelia generally have a greater fat concentration and are more likely to survive in the cold. Are there any other microbes that you're aware of that have an impact on the survivability of either the cold in the winter or the heat in the summer, or just make them more resilient in their environments?
I'm not familiar with any microbes that have that direct impact on their tick vector host. But I do know that ticks themselves have different susceptibilities to things like temperature and humidity. So the Lone Star ticks require a certain amount of heat, and they do not do well in our very cold Minnesota winters. So that's one of the limiting factors. So the ticks themselves can definitely have different features, just like other animals. Some are more adapted and can survive cold weather, hot weather, humidity, etc.
Yeah, I can certainly tell you that when we have mild winters, we have many more loans and they're much more aggressive. When we have
Yeah.
colder winters, that generally does tamp down the Lone Star tick population, and it does. We generally see less of them, or at least I see less of them coming at me. For
uh,
sure.
yeah, I think you're right, Rich. And because of that, it is worrisome what's going to happen with climate change as we're seeing milder winters. We're going to see probably more ticks that survive the winters. There's going to be more ticks that are out and about looking for their next blood meal earlier as the snow starts to melt and extend later into the season. So it's going to have an impact for sure because of those temperature differences you mentioned.
I think Rich froze.
Yeah.
So
I
that
think
means
Rich.
I get to jump in here, Dr. Pritt. I've been waiting, so this is wonderful.
All right.
So let's talk a little bit more about not only when these ticks are most prevalent, I know that this cool stuff we're talking about now, but I have so many questions about things you've already discussed with Rich. The first one being you talked about earlier on, Dr. Pritt, that you in your early career would identify these atypical triggers. And I'm paraphrasing, but you basically were saying, hey, we did
Yeah.
some laboratory tests. They didn't hit as an exact match for an existing Borrelia species. So we had to dig deeper. And then we discovered this species of the bacteria, whether it was Borrelia or whether it was Ehrlichia. And then you named it because you discovered it.
Right.
I guess my question is that something unique to you in your lab because you pushed harder and other labs would have discarded that as being a negative test? Does that make sense?
It does, Matt. And, there's multiple components here. So, first of all, do you have a test? I'm just asking as a hypothetical question, that this would have an impact of on a lab if they have a test that's capable of detecting an atypical result, something that was out of the ordinary. So if they have just a test that gives a black and white answer, then no, they are not going to detect it if it's not exactly what they're looking for. Our tests were broader, multiplex, detected multiple species, use this special kind of technology called melting temperature analysis, so we could actually see a different temperature. Okay, so that's step one, having the test. Step two is, I guess, having the curiosity and the drive to follow up on it, because another possible outcome of our discovery could have been initially that those two technologists that saw the atypical result, they could have just signed it out as anaplasma atypical and left it at that, and no one would have dug further. Now, eventually someone would have noticed this, and maybe 10 years down the road someone would have discovered it. But a lot of times discovery is being in the right place at the right time, but then having the willingness to ask questions and investigate. And then for me, I would say it really emphasized the importance of teamwork, because our lab does great science, but we're a clinical diagnostic lab. We focus on diagnosing patient specimens. We don't usually go out into the field and drag for ticks, for example. I've done that every once in a while for fun, but it's not my primary job duty. So I had to team up. I had to team up with various people. We also don't grow these organisms in culture. It's really tricky to grow them in culture. It's really just people that have research labs that do that. So I had to team up with people from some of the universities and the CDC. And so that would be another step. You have to be willing to pick up the phone and talk to someone and say, hey, I have this thing that I think is atypical. And then, make sure that they realize that you're actually doing real science and show them the data, and then hopefully be able to make that connection.
So Matt, can we just walk it back for the folks who are new to the Lyme community to talk about the different types of testing? So, Dr. Pritt, can you first share with our folks what is the difference between the direct and the indirect testing and the different types of indirect testing that exist?
Yeah, so a direct test is something that directly detects the organism or a part of the organism. I talked about PCR. That's a direct test. You're detecting the DNA or RNA of that organism. Culture is also a direct test. Antigen detection methods, those are direct tests. So for everyone in the COVID world, you look back to COVID, those rapid antigen tests that you buy over the counter at the local drugstore, those are direct tests. Um,
Can
in
you describe
Oh,
what an antigen is,
yeah.
and what a culture is?
A, yeah, let's start with the easy one. A culture is that you could actually help the organism grow. The organism itself is responsible for growing, but you're providing the right nutrients, the right temperature, the right environment so that it can replicate itself. That could be bacteria, fungi. Think about, okay, let's say you decide to leave a loaf of bread out for 2 weeks. That basically is like culture for the mold that's probably going to grow on that loaf of bread. In the laboratory, we do very special types of culture media that have been optimized to help the bacteria grow. In this case, I partnered with Dr. Uli Munderloh at the University of Minnesota because she specialized in growing up Ehrlichia species. And so she had this very special media that these bacteria just love, and you give them heat and nutrients and they replicate themselves.
Okay, so
Um,
that
yeah,
would be in a petri dish
it's
culture.
like it's in a liquid media, so it'd probably be in a tube or a flask,
Okay,
but same concept. You could grow in a petri dish as well on like an auger. So that's
okay,
culture. Now, antigens are usually a protein that is on the outer surface, or it's something that your body forms an immune response to. So antibody, antigen. Um, the antigen is something that could be detected when you're talking about an antigen in a laboratory test. So it's not the DNA, it's a protein that we're detecting from the organism. Now,
okay.
usually you could have one strand of DNA that might then go and make hundreds of proteins, antigens. So sometimes those antigen tests can be very sensitive because there's a lot of antigen present.
Okay, so those are the direct tests. Now, what
Yep,
is the
those are the
indirect
direct.
test?
Now, the indirect tests are measuring the human's host response, or well, the host, in this case, a human, uh, antibody response. So now we're talking antibodies. All of us as humans and all animals, all creatures have some sort of defense mechanism to help protect us from harmful pathogens. And it's called our immune system. And as part of our immune system, we produce these things called antibodies, which attach to foreign proteins, antigens, and then kind of triggers reactions that allow us to destroy them. So your antibody response is a good thing, and it helps us get rid of things that shouldn't get into our bodies. So we can measure the antibody response because if you detect an antibody response, that means that person had seen that organism. Their body had encountered it and had produced that antibody response because antibodies have to get produced. It's a form of immunity called adaptive immunity, meaning right now, I probably don't have antibodies to Ebola. That's a good thing. That means I've never been exposed to Ebola. But if I ever got Ebola, my body would form antibodies, and then someone could do a test on me and detect those antibodies. I use a pretty
Oh,
severe example because I was trying to think of
yeah.
something that most people probably wouldn't have. So if I were to test you, Rich, for antibodies to Lyme disease, I don't know if you had Lyme disease before, you would probably be positive for those antibodies. The test would be positive for Lyme disease antibodies because once your body forms them, they don't go away.
Okay, one more piece before I hand you back to Matt. I know
Yeah,
he's
yeah,
anxious,
sure.
but again for the foundational conversation. So, how do the different direct or indirect tests indicate what type of microbes I'm harboring when you put me through these various types of tests?
Yes. Okay. So this is a little complicated too, but this is good. So first of all, it takes a little while for your body to form antibodies, 1 to 2 weeks. So if I am sick on day one, an antibody test is probably not going to be positive because my body has not had time to form those antibodies. So that indirect test is not good in that early window of time. Whereas a PCR test is probably perfect in that early period, or a culture-based test, or an antigen test, because it's a direct test. It's detecting the organism. As long as I'm detecting or testing the right specimen where the organism is actually found. Now, I forget the question you asked, though.
So how do these different tests give my doctor, or how do you as the pathologist who's testing my fluids, and we'll get into that piece of it in a minute. How do you as a pathologist now determine in the report you're going to send back to my clinician what types of microbes my body is
Mm,
harboring?
right. Okay. So an indirect test, the antibody-based test, it doesn't matter what type of organism your body's going to form antibodies against it, whether it's a bacterium, a virus, or a parasite. So the way the test is developed, though, the test will specifically detect antibodies to, let's say, Borrelia burgdorferi or Babesia microti, and they're different tests. So it's the same principle regardless of the type of organism, but the tests we use usually will detect a specific organism and maybe some closely related organisms. PCR tests are more specific. They're looking at a specific organism. They'll detect the DNA or RNA. When we get into different types of organisms, things like viruses can be RNA viruses. So you have to make sure that your test detects RNA instead of DNA.
Okay. And are different types of tests more or less effective based on the type of fluid that's being tested? So, for example, are some tests better testing urine versus blood versus spinal fluids? I mean, is
Yes.
there a difference in the effectiveness based on the type of fluid that's being tested?
Yes, but it's not exactly the test. It is where is the organism. So let's say that you're going to test for Lyme disease and you are testing, I'll just say toenail clipping, something crazy. You're probably not going to detect the pathogen because that's not where the pathogen lives. In fact, Lyme disease, Borrelia burgdorferi, is a tricky one because it's not found in high numbers in the blood either. So if I test the blood with a PCR test for Borrelia burgdorferi, I only have a 50-50 chance of detecting it during the early stage. But if I test a skin biopsy where there's a bullseye rash, I have a pretty good chance of getting a positive result. Now, it's true, some tests are designed to work better on different specimen types. Some tests work better on blood or urine, but really what's going to be the most important is where the organism is actually present. We know that Borrelia burgdorferi, for example, is just not found in high quantities in blood or in CSF. So those are not specimens that are going to give you a high yield by PCR, at least.
Okay, so now we have the foundation, Matt. I apologize. I wanted to make sure we get that.
No, you're
You can
Rich.
now.
You've inspired other questions about what you're saying. So I'm going to go even
Okay.
further now on that before I
Okay.
go back to my other question, which is even deeper. So thank you,
Okay.
Dr. Pritt. So I'm going to recap and tell me if I'm wrong. And please don't be kind. You can tell me I'm wrong as I'm recapping your discussion with Rich. I want to make sure I have a good
Okay.
framework of these things for our listeners. So in the direct testing category, we talked about PCR, culture, and antigen testing. Is that correct
Yes.
so far? Okay.
Yes.
And the PCR testing is looking for DNA or RNA of the bacteria and or virus to
Yep.
directly detect it in your blood, but it's limited because when you're looking at blood or CSF, Lyme disease specifically is not highly concentrated, right?
Yes.
Okay.
Now you said blood and CSF, but depending on the organism you're trying to detect, there's other specimen types too. But yes, everything you just said is
Okay.
correct.
Which is excellent because, and then we also talk about skin biopsies. There's been a lot of research about testing with skin from other researchers that, these horror stories with patients that the skin biopsies have identified the bacteria after so much suffering. One of the ones we've heard a lot about is urine testing, and we're going to call them out. You don't have to mention them by name if you don't like to, but DNA Connections does a lot of urine testing. We can't do that here in New York State where Rich and I live. And a lot of people swear by DNA connections, and other people that are researchers and pathologists have told us off the record they're horrible, their results are skewed, never recommend them. So we've heard such extremes. So you don't have to mention them specifically, but when it comes to urine testing using direct testing, what are your views with that model?
Well, first of all, I'm not familiar with that lab, so I can't comment on their testing. Although there are some labs out there that are doing very unconventional tests that are not really recommended, and you wonder about the science. But I will say, in terms of urine, we have not had good experience with that being a good source for detecting Borrelia burgdorferi for Lyme disease. Now, urine is actually a pretty good source for detecting something like West Nile virus by PCR. It's actually shed a little longer for a longer period of time. But Borrelia burgdorferi, at least in my experience, has not, and it's not recommended by the CDC. Urine has not been a good source.
Okay. So then in addition to PCR as being a type of direct testing, right, either using skin, blood, urine, CSF as the modes, the other one we said was a culture, which is basically where you take a sample, maybe blood, maybe whatever, right? It could be, I guess it could be anything. Could it be any of the median, skin, blood, urine, and CSF for cultures?
Yep, you could try all of those. I don't know about urine, but the specimen types that have been tried would be CSF, synovial fluid, skin, and CSF. Those are the big ones.
Okay. And that basically means you put it in this laboratory environment and you put it in this essentially culture that is allowing it to grow. So a good temperature, you feed it nutrients, and you let the bacteria or virus grow to prove that that was active in the patient. And if it grows, then it's a positive test. That's the general idea, correct?
Yes, and it has an advantage over PCR because if you detect something by PCR, DNA or RNA, that just means that it's there, but it doesn't mean it's still alive. But if you grow it up in culture, there's no question it's a live organism.
Got it. Okay, that's a really important note. Thank you for sharing that. So then we have antigen, which essentially, as you mentioned earlier, and I love the way you described it, you have your antibody, which is your immune system and your body protecting you, which mounts a response against an antigen, which is something that's a protein on the outer surface. They call it the OSP, right? The OSP, the outer surface protein of Lyme, for example. The antibody attaches to that, and then it kills the thing. But your direct testing is looking for the antigen of the bacteria, the virus, whatever it might be from a tick-borne disease. So that's a different type of direct testing, correct?
Correct. It's kind of like PCR. It's just usually not as sensitive as PCR, but you are basically detecting the antigen, just like the PCR is detecting the DNA or the RNA.
If there is an antigen, does that mean it's actually alive, unlike a PCR?
You have no idea. Nope, it doesn't mean that. Yeah, so like PCR positive or antigen positive, it does not mean it's still alive. Only culture is really going to show you that.
Okay.
Or you can do some transcription studies, but we won't talk about those because no one's doing those really.
Okay, so put that off to the side. So one of the things I found fascinating while doing research on you is that I'm going to butcher it. Unlike Rich, I'm going to go for it. Borrelia myonii, which I know I butchered. Your discovery of that back in 2016, one of your discoveries was that it appears in much higher concentrations in the blood and the body than Borrelia burgdorferi. So first off, you have an idea why that is. And also, I think that gives us a better advantage in testing because it's highly concentrated, right?
Right. So yes to all of those. Borrelia mayonii. If you're trying to remember how to say it, think of Mayo, like Mayo Clinic. Like we named it after the Mayo brothers. So mayonii, you're right. We found it in higher quantities in the blood. So for Borrelia burgdorferi, the bacterium that causes most Lyme disease in the United States, blood is not a good source for PCR. There's just not a lot of the organism in the blood. It doesn't mean the bug's not in the body. It just doesn't happen to be in the blood in any high quantity. But that doesn't appear to be the same for Borrelia mayonii. And that makes blood a much more attractive and effective source to use for PCR. It's a little tricky though, because Borrelia mayonii is not very common. So is it cost effective to do this PCR test on every single person? Probably not. And we also know that the indirect test, the serology, will be positive with Borrelia burgdorferi and Borrelia mayonii. So most people probably just get the serology test and don't get the PCR.
Okay. So when you say serology, does that mean indirect?
The indirect, yes.
Okay.
Looking for the antibodies. The two
Okay,
are
so I'm
so,
gonna.
yeah, they're so similar, Borrelia mayonii and Borrelia burgdorferi, that if a person is infected with Borrelia mayonii, they'll be positive for the serologic test, the serology for Borrelia burgdorferi. There's cross
Okay,
reactivity.
so I definitely want to go to the antibody piece in a second, but I want to dig
Okay.
a little bit deeper because something else you said was really fascinating, which was if we test the skin where the infection is, we're going to have a high chance of a positive test. And I don't hear a lot of people doing that. I read studies and articles of researchers that are doing it now, but where I live, I don't hear that much. I don't think I've never heard that where I live locally. So the question is, if they have a bullseye rash, does that mean the infection is heavy there and we should test the skin? And is that a good recommendation if somebody happens to be lucky enough to have a bullseye rash?
Well, I wouldn't necessarily recommend it. So, first of all, if you are in a Lyme endemic area where you know there's Lyme disease and you know it's a pretty high rate and you get a bullseye rash, that is considered diagnostic of Lyme disease. You should just go to your doctor and get treated. Um,
Meaning you have Lyme disease, meaning you don't need a
yeah,
test. You have it, get treated, move on, right?
exactly. Move
Okay.
on. You don't need any lab tests. You don't need to pay money to get any testing done. Just go to your doctor, show them the bullseye rash, show them the picture, and you'll get your antibiotic. Because remember, as we said earlier on, early treatment's key. You want to get your antibiotic as quickly as possible to decrease the risk of having complications.
Right.
Now, let's say that bullseye rash is not typical. It's not a bullseye. Maybe it's really faint and kind of atypical and you're not sure. Okay, you could biopsy it. The reason people don't, though, is it's invasive because to biopsy it means I have to cut a piece of your skin out of your body and bring and send it to the laboratory. And if you live in an area, Rich, you were talking about that you've been bitten, it sounds a fair amount. If
He's a
every
tick
time
magnet. Yeah, he gets bit a lot.
so if every time you got bitten and you get a little red spot and then you had to go to the doctor and get part of your skin cut out, that probably wouldn't be very popular.
He'd have no skin left if that were rich. Yeah, he'd be skinless.
Yeah, I hopefully you can come up with another test, maybe some kind of a swab that'll make it easier. So anytime I have a rash, we can test it without
Mhm.
being gouged.
Exactly. So that's why it's not that common. It's a good test. It's much more likely to be PCR positive if you test the skin by doing a biopsy. It's just it's invasive. It's a lot easier on the patient to take a blood sample. It's just
Got
that
it.
the blood is not going to probably be positive because that's not where the organism is.
Right, and it's too early potentially, and so on and so forth.
For
And it's not.
serology, yeah.
Yes. So before we go on to that piece, I want to talk about the rashes in general because it was about, I think, a month ago, the JAAD, right, the official dermatology journal of the American Academy of Dermatology, put out a study that I don't think got the attention it deserved, but we pushed it, and a lot of people that follow us were really engaged by it. That the traditional bullseye rash is flawed, and this study basically said it's not always a bullseye rash. It
Right.
can present like a Christmas tree, and there's some photos out there, which is really fascinating to see and other types of rashes. And they describe it pretty well. And they're saying the likelihood of a bullseye rash is not as common as you'd think. And there are different types of rashes. So did you read that journal article? And what are your thoughts on that?
I'm familiar with the research. I don't know if I read that particular article, and there have been others. There have been other people that have published good studies on this. You are correct that classic expanding bullseye rash is probably not the majority of what people see, or at least there's a substantial number that are not that picture perfect view. And even the CDC website says, the erythema migrans rash doesn't always have the clearing and it should be an expanding lesion. That's one of the diagnostic criteria, but it may not look like a bullseye.
Yeah. Yeah. So let's talk about now the indirect, right? Let's go there. And because on the indirect side, this is where I think it gets interesting. So the traditional two-tier testing, right? And there's the ELISA, I believe, and then the Western blot. And now I'm kind of going way back. Can you describe, I think they're both indirect tests, correct? And how are they different?
Yes.
And are they different categories? If you can help us understand that a bit.
Yeah, so the ELISA, that's the first test usually in the traditional or standard two-tiered algorithm. So you would do an ELISA, which is pretty sensitive and is good at kind of screening, but you're going to have false positives. So then if if the first test, the ELISA, is positive or atypical, you follow it up with a second test, which has better specificity, meaning it's not going to give you a false positive. And if the second tiered test is positive, then both tiers, the 2 tiers, are positive, and you get a result of positive indirect test for Lyme disease. And the second test used to be a Western blot or an immunoblot, but we now have the modified 2-tiered test, which is a little bit more sensitive, and that is 2 ELISAs. 1 ELISA, and if the first ELISA is positive, then you do the second ELISA, and then if they're both positive, then you say, yes, this person has Lyme disease. Now, to answer your question, Matt, whether it's an ELISA or a Western blot, it is still an indirect test. It is still looking for antibodies. It's just a different way of looking for antibodies.
Yeah. Is the second ELISA the same test, or is it a different version of it? Or is it literally the same thing, just validating the first one?
It's a different test looking for different antigens. So if the first test is looking for a certain number of antigens, and then the second test may be looking for a different combination of antigens, or it's just a different manufacturer, you're basically using 2 different tests, but you're still looking for antibodies at the end of the day. And so, as we had said earlier, when Rich and I were talking about this, it takes a good number of days before your body develops antibodies. And so if you test on day 1, you're not going to have antibodies. That's the major limitation of these indirect methods, is it takes a good week for your body to develop antibodies. So that early stage of Lyme disease, when maybe you have a bullseye rash or an erythema migrans rash, you will not have antibodies probably.
Right. So I think there's been a lot of controversy around these antibody tests that we've heard, at least from the community. So, for example, I believe it was in the 90s when the first vaccine came out for Lyme, the standard testing prior to that was looking for, I'm going to make up a number. Well, I won't even make up a number, a broader range of antibodies. Then after that, the number of antibodies they were looking for went down, which meant we had less positives. But a lot of people said that's flawed because we're not capturing the people who are really sick, because we've changed the criteria by looking for different antibodies. Now, I get it's very complex, and a researcher and a scientist like yourself, it's something you have to deeply understand and study. But can you try to distill that for us and explain to us what that means, right? A different number of antibodies, and what that actually translates to in, like, if you're explaining it to a 4th grader, and how that makes sense.
Yeah, I'll do my best. I'll say that the antibody testing is really done by my colleague, Dr. Ellie Thiel here at Mayo. I focus on the molecular testing, but of course I know the basics. So it's my understanding that the earlier tests detected a whole number of different antigens. Some are very specific for Lyme disease. Some were actually found in people that never had Lyme disease. So there are some antigens that just happen to be found in normal, healthy people. So you'd be getting a lot more false positives. So it's my understanding what they did is they narrowed down the antigens. You're right, they did decrease the number of antigens that were used in their antibody tests, the number of different proteins that the antibody test detected, because they were focusing in on the ones really associated with Lyme that were the most specific for Lyme disease.
Got it. Now, when you're saying you're looking for the antigens, I'm getting a little confused, because we talked about a direct antigen test, and now we're talking about an indirect antigen test. So are we looking for the antibody in what you're saying for the indirect, or we're looking for the antigen on the OSP for the direct, or am I misunderstanding that?
I might have said it confusingly, but with an antibody test, you use different antigens to detect antibodies. Remember, we talked about
to pull them in and attract them to see the antibodies.
Yeah. So say you have an immunoblot. It's basically just a piece of paper, nitrocellulose paper. And on that little piece of paper, you put antigens on the paper. Then you take your patient's blood, their serum, and you apply it to that piece of paper. And if they have antibodies, it will bind to the antigen. And then if you use a little color method, you get a little line, like the COVID test that you take at home that you buy. You get a little line because you have the patient specimen has the antibodies, and it detects the antigens. And now you have this little sandwich.
Right, like the antibodies running to the antigen and attaching to it, and then it's lighting up for you to know that it's happened, right? Oh, that's interesting. Okay.
Yep. So yeah, you kind of have to talk about both the antibodies and the antigens you're using. So you were talking about the different antigens. The antibody tests initially tried to detect a whole number of different antigens, but some
Yes.
of those just weren't as useful because normal healthy people would get positive results.
So
So
what I'm taking away from this, Dr. Pritt, is that we've had so many people tell us testing has come a long way. We're gonna have a test that's definitive. You have Lyme disease or you don't. And look, Lyme disease. What is Lyme disease? Obviously, Lyme in the most scientific fashion is Borrelia burgdorferi. We don't really believe that. In our community, chronic Lyme disease, chronic Lyme disease is many infections, right? It's probably co-infections, it's other opportunistic infections, parasites. When somebody gets as sick as I did, and a lot of people in our community, it's a lot of things making us sick. So I think that's a misnomer. That's a problem as well. It's not just I'm looking for Borrelia burgdorferi, I'm looking for other things. But even just Borrelia burgdorferi, there is no magic test, and there are pros to direct, and there are pros to indirect, and there are going to be just statistical odds in your favor that may or may not, definitively give you an answer. And I think you're being very honest about where we are with science today, that we're not there or close to being there to get 100% accurate tests for all the various species of Lyme and co-infections. Is that a fair statement?
it is, Matt, and you kind of hit the nail on the head. Like, it's not perfect right now. We didn't really get into this, but the indirect tests, as we said, it takes a while to get a positive result. Your body, it takes, a good week to form antibodies. That means that Lyme disease serology, that indirect test, it's not a very good test for the first several days of infection. You could try PCR, but for Borrelia burgdorferi, we know that it's not usually found in the blood. And unless I gouge a piece of your skin, it's not very good to do PCR on skin either. So it's that early window we don't have a good test for. Now, if you have a bullseye rash or an erythema migrans expanding rash, and you're in a high endemic area, maybe you don't need a test. Maybe you just say, you know what, that is indicative of Lyme disease and we're going to treat you. And that's probably the best thing. But what if you're one of those unlucky patients that had a bullseye rash on their neck and you didn't happen to see your neck that day, and you totally missed it, and you don't have someone living with you that said, hey, Matt, what's that thing on the back of your neck? So you can imagine you might have had erythema migrans rash and it could have been missed altogether, or you could have been one of the people that actually never had an erythema migrans rash. So there's that as well. So in my mind, there's two major gaps, and I know I'm probably jumping ahead, but there's two major gaps in our testing for Lyme disease specifically. And the first is that early window. We don't have a good test for that early window, that first week, 7 days or so, where your antibodies haven't yet formed for the people that don't have a nice identifiable erythema migrans rash. Now, the second gap we can get into later, but that's when you have longstanding chronic symptoms and we don't understand what's actually causing it. We need to be able to understand that as well.
Right.
So Matt, I think we should just, for the purposes of the folks who are new to the testing conversation, who are listening to this podcast or watching us, is that the testing that the typical person is taking after a tick bite, or they have symptoms that clinically indicate that they may have Lyme disease, is a set of tests that are going to be antigen based, that are going to have limitations based on the choices that the testing professionals are making. So, for example, the average person is going to have a tick disease panel from a Quest or one of the general labs, and what we're definitely not going to see is a test that will have a Borrelia myo, my,
Mayonii.
Mayonii. I knew
Mayonii,
that.
even though I might have Borrelia mayonii, right? Because that antigen is not going to be on the test, and therefore, even if I have developed a antibody to respond to that microbe, it's not going to test. I'm not going to test positive because it's not going to be a part of the tick panel.
Okay. Yeah. So yes and no, but mostly yes. So it is true. Borrelia mayonii not a very common pathogen, as we talked about, or at least we don't think it is. So you're right. Most people don't have tests for it, and there is no FDA approved test. And there are a lot of laboratories that only want to use FDA approved tests because it's a lot of work and it's very challenging to create a laboratory developed test. So you're right, Rich. There's a very good likelihood that the tests that people are using probably won't detect Borrelia mayonii if they're doing PCR specifically.
So, but
Now,
the right. So, but let's stay with the antibody testing because that's what's going to be a majority of the tests
yeah,
that the average person who's listening to this podcast is going to have to receive, right?
right.
So, on the one side of the coin, one of the flaws that we're going to have is there are going to be a number of different microbes that we may have been spit into us from a tick bite that are not going to be testable because the antigens are not going to be there for our antibodies to respond to, correct? So that's one flaw.
Yes, although I will say Borrelia mayonii appears if you're infected with Borrelia mayonii, you will probably be positive with the standard Borrelia burgdorferi
Okay.
indirect test.
Okay.
But it's only good for test for Borrelia burgdorferi, Borrelia mayonii. If you have a Babesia infection, it's not going to detect that.
Right.
So,
Right. So
yeah, what is
the
ordered?
spectrum of
Hmm.
microbes that is going to be testable is going to be limited. So let's start with that one piece, right? So
Correct.
we
Yes.
agree. There may be some species of some microbes that may test positive, or may give a positive indication that would allow you to look further if it's a species of something that's going to be tested, right? So we have that problem, right?
Hmm.
But we have a second problem. The second problem is going to be the timing, right? Depending
Right.
on when you're going to be taking the test, if you're taking the test too early and your body has not provided an antibody response, you're not going to test positive for something that your body may be testing. You may be harboring, right? But then we
Yes.
also have the other situation, which is we may have been bitten in the past and we may have antibodies from the past, and those
Hmm.
antibodies may show up today, even though our body has already cleared those
Yes.
microbes, right? So we could test too early, we could test too late, and there are a
Hmm.
lot of different problems with if an antibody does appear, correct?
Yeah. Yeah, you're hitting all of the main points there, Rich. Antibody tests, our indirect tests, are flawed. In the early period, they may not be positive because it's too early. Your body hasn't formed antibodies. And then once you have been infected and developed antibodies, you usually have those antibodies for life. And so if I were to test you next year, you're probably still going to be positive. But if you had been treated, that doesn't mean it doesn't necessarily mean that you got infected again. So it's tricky. That's why that's why in my lab I really like the direct methods and
Okay.
I don't do the indirect methods, but they are still considered the gold standard. And
Yeah.
my colleague, Dr. Thiel, we work closely together. She oversees the indirect methods. I oversee the direct methods. You do kind of need both together. It's
Right.
just I prefer to work with the direct methods because if I have DNA or an antigen or I grew it in culture, I know it's there.
Okay.
Right.
So that's
But
let me. I want Matt, if I could just focus on
yeah.
one more piece of the indirect testing, just so that folks have this in their quiver when
Mm,
they understand how the testing can be flawed and how they could either have a positive that is not helpful or a negative that is not helpful, right? And the other
Right.
piece of it is one of the things that I learned at the ILATS conference when I was conducting interviews there is Dr. Dempsey shared with me that one of the reasons she does not use indirect testing is not because of the two issues that we've identified here today, but she said there are going to be some cases where you're going to have a positive antibody response to a microbe that doesn't exist if you have mast cell activation syndrome,
Oh,
or you may have no antibody response to a microbe that is there. So not only is the test flawed because the timing is significant,
mhm.
or the antibodies the antigens that the test is making available for the antibody response, but your body maybe producing antibodies to something that doesn't exist because of um, challenges with the immune response, or it may
Mhm.
not respond at all, and you may not have antibodies at all. So, I mean, it just seems to me that it's just a mess when you're looking at indirect testing
Yeah.
for a whole host of reasons.
Yeah, and I'll just elaborate on that, Rich. So, people that have like rheumatoid arthritis or their immune system has kind of gone into hyperdrive, or they have a condition where they have mast cell or plasma cells producing antibodies like multiple myeloma, you're going to get false positive results. We know those indirect tests have that possible or potential where, yes, you're going to get a positive result and that patient doesn't, it's a false positive.
Right.
We also know that if you are profoundly immunocompromised, your body cannot produce antibodies. Let's say you have advanced HIV infection and all of your little B cells that produce antibodies are dead.
Yeah.
You are not, yeah, or are no longer activated because yeah, they're not getting the right signals because of the virus infection. You're not going to have antibodies to detect.
So when the standard for Lyme disease testing is indirect testing, right? The two-tier test is indirect testing. And you have timing issues, you have a immune response, whether it be autoimmune or non. Um, and we have choices that are being made by the testing companies about what antigens are going to be used to test or not. We really can't rely on this at all, right? We have many people who have had false positives and many more people have false negatives. And we have a whole community of people who are sick and looking for validation for their disease. And the testing protocols that we're using are so flawed that in some cases I could argue it's better not to do the test than to do the test at all.
Yeah, what I'll say is clarify a little bit. As I had mentioned earlier, the one area that I think there's a big gap is early diagnosis. For we don't have a good test for that, and you're right. The serologic tests have, early on for acute disease, it might miss half the cases. We do know though that after that initial window, the serology, the indirect tests are actually pretty good in detecting. So by the time it's past like week 2, most people that have Lyme disease will have a positive result. But you're correct, it is an indirect method. It's still looking at the antibody response your body produced, and it's probably going to stay positive for a long time. And so if you get reinfected, a positive result may not be meaningful. So I think that's why it's so complicated, Rich. You're right. That's why we say if you have an erythema migrans rash, don't go get testing because it's probably early on and you're not going to have an antibody response that's positive anyway. Just get treatment. Make sure you go and get treatment right away. Uh, we also know that if you're going to do a test in that early period, you should wait and do another test later on because that first test might be negative. That's why we have all these complex testing algorithms. In our
So,
lab at Mayo Clinic, what we do, we first of all say if you have an erythema migrans rash, no testing's needed. Go get treated. We want you to get treatment. Um, but if you don't have a classic erythema migrans rash, we do recommend the serologic testing, the indirect, and we also recommend a PCR panel for all the other things, Babesia, Anaplasma, Ehrlichia, to make sure we don't miss any of those as well. And then it may be indicated to get a follow-up or Lyme disease serology. So it's algorithms. You're right. We need a magic bullet test that can detect
yeah,
everything right off the bat instead of these complicated algorithms.
let me share one last experience with you before Matt revisits or moves forward with his thought process, which is one of the things that motivated me to participate in this podcast experience is that I was bitten by a tick, and I actually saved the tick. Matt actually recommended I save the tick. I brought the tick to my doctor's office. They said there was no need to treat it, no need to test it. In fact, it was a Lone Star tick, and it was a female
Mm,
Lone Star tick, had a spot on the back. And at the time, I didn't know what that was. And when I
mm,
was showing that to my healthcare professional, what he said was, I don't need to look at the tick. The tick is not significant. So
hmm.
6 weeks after my initial visit, I go back to the doctor's office. They test me for Borrelia, right? So And of course, the test comes back negative. I had no antibodies in response to that. Shortly after that, we start our podcast, and we interview a tick testing company, and the tick testing company agrees to test my tick as part of the process of us interviewing her. And when she tests my tick, it was positive for Ehrlichia.
Yeah.
But I was tested for Ehrlichia, right? It was not even a part of the panel of tests. So, there are choices that are being made by our medical professionals that are not broad enough for them to determine whether or not, you are harboring those microbes. And because there are a diverse set of microbes and there are a diverse set of treatment options for the diverse set of microbes, one of the things that I'd argue to you is even if someone has a bullseye rash, they should still be tested because there's a whole range of other germs that they could be harboring that would require something other than doxycycline in order to be able to properly treat them.
So I like what you're saying, Rich, and it goes back to what I was saying about an algorithmic approach with a panel. So we recommend Lyme disease serology because it's, unfortunately, for better or for worse, it's what we have. But we also recommend a PCR panel that detects Borrelia miyamotoi, Babesia species, Ehrlichia, and Anaplasma species. So you're kind of throwing out a broad net there. Now, we didn't get into this, but I would like to see in the future an even broader net, but not by adding on multiple tests because they get expensive and our poor patients have to pay for these, and each PCR test can be $100. And if you're doing five, $500 every time you get bitten by a tick. So what I'd like to see is a test that's fast, that is what we call a metagenomics test, capable of detecting anything that person has, be it a virus, a bacterium, or a parasite. So my lab's working on that now, and that's what I'm most excited about, is that ability to detect everything with just a single test with a single blood draw.
Okay, so
Oh, we got to get there in a second, though. We're not there yet.
Okay,
because we're gonna get there.
go ahead,
I'm
Matt.
jumping ahead.
Yeah,
Okay.
no. I just want to close a couple of loops here because Rich, of course, and you and Rich are bringing out a lot of good things. But one of the things you said, Dr. Pritt, that I want to just question is, you said that indirect testing for Lyme specifically is not great early on, but it's pretty good later on. But we have observed from doing this for about 5 years or so that a lot of people that come to us have been sick for many, many years. They're bedbound, they're really, really unwell, they're having a host and a wide range of symptoms, and oftentimes they're immune compromised. And then they finally hear about Lyme, they get tested with the indirect testing, and it's not positive. But after the second or the third or the fourth, it is, which makes me think, is it maybe, timing wise, like right in the middle of the sandwich, right? Like it's where like the meat would go in between two slices of bread, where early on, not so good. Okay, maybe I'm 8 weeks in. Yeah, it's pretty good at that point, right? But now you're a year or 2 years in. Are you immune compromised from the infection, and therefore you're not mounting a proper immune response where the testing becomes less effective, and now people are struggling to get a positive diagnosis? Or do you think that's not so much the case?
I don't think that's probably the case, because the studies where you have Lyme disease, you could grow the organism. These are people that definitely have it. They've shown that there's that early window where you get a false negative, but then in the later stages, it's detectable. Second stage, like disseminated disease and neural borreliosis, Lyme arthritis. But there's so many unknowns, like, these people that are suffering and they got, 4 tests and it was maybe only the 5th test that was positive. What I don't know is, are they truly positive? And I'm not questioning the result or what they actually have, but if you do anything over and over again, there's always false positives. So if I run a test and I run it again and I run it again, I'm eventually going to get a false positive. That's a known limitation of all laboratory tests. Let me put it this way. We have a test, if it's 99% specific, that's pretty good, right?
Yeah.
That means I will get 1 false positive out of every 100 tests I run. So if I run it enough, I'm going to hit that false positive. 99 out of 100, it'll give me the correct result. But 1 out of 100 will give me a false positive. So I don't know if that's the case with those people that you're talking to, but I guess that is something I would raise is I do get cautious if you're just running the same test over and over and over again and you suddenly get a positive. That's a little suspicious to me, especially if it's the same test. If it's a different test, maybe it's a better test. But if it's the same test you're running over and over again, I'd be a little suspicious why now all of a sudden it's becoming positive.
Now, this kind of ties back to what we were talking about earlier, which was the atypical side of things, right? Depending on the lab, is it possible if you have like a cousin of what you're testing for, right? A different Borrelia species or a different Ehrlichia species, that you're less likely to pop, but you maybe will 1 out of every 4 times because it's maybe an unknown species or an unknown type or a type that we're not testing for? Or is that not really a thing either?
I haven't really seen that. If you're going to have cross reactivity from something that's a novel atypical result, you'll usually find it right away. Like there's,
You'll find it with the test for the one that we know about. You're saying, right? It'll pop.
Exactly, and it'll pop right off the bat. You'll get a positive, or might not be. You could run it 3 times. It'll be positive 2 out of 3. Like maybe present really low levels, but it wouldn't be something that you'd have to run 10 times or 5 times before you got a positive. You'd usually see it right away.
Got it. And this is something I've been thinking about for a good portion of this interview is, you've touched on Ehrlichia, Borrelia, Babesia, right? All the different types of tick-borne diseases, but then subtypes. Now, is that the proper terminology? So like, what is Borrelia versus Borrelia burgdorferi, Miyamoto, I'm going to butcher it again, Mayoni, whatever it is.
Oh, Mayoni, yeah.
Mayoni, and so they're different subtypes. And then what is the real world impact? Do these subtypes have drastically different symptoms? Because we've heard that different subtypes of Borrelia might be more prone to be neurological, whereas other subtypes of Borrelia might be more prone to be, inflammatory in your joints. So, is that something you found as well? Because it's interesting to see that, it's Borrelia, but these different types could be more severe or less severe.
Yeah, there's really good data to show that there are certain subtypes, that's a good word, or genotypes, because it's part of their gene. It's a slightly different than some of the others. Their genotype, some of them are associated with worse disease. They're more likely to be neuroinvasive or more likely to cause Lyme arthritis or more virulent disease. Yeah, and that's true. We do not, in my lab, test for those different subtypes because the treatment's still the same, but it's something that metagenomic test I talked about that could detect everything. It would have the potential to detect the subtypes as well.
Got it. So you're looking for the main, not the sub. And if you have the main, the treatment's going to help you with all the subs. Is kind of what you're saying, right?
Well, at least that's what the standard is. Yes. At this point, even if you have one of those more virulent species, that means maybe you're more likely to have Lyme arthritis, but it doesn't mean you're going to 100% get Lyme arthritis. So you're still going to be treated the same right now. Now, in the future, though, maybe subtyping will be important if we have a different treatment that's more aggressive or better. But that's, there's no data to support that right now.
But what about with Borrelia, like neurological Lyme versus just rheumatological Lyme, right? Wouldn't you need a different type of treatment to better get into the brain to treat the Lyme in the brain versus the Lyme in the joints? So wouldn't that subtype that's more neurologically inclined matter to dictate a specific type of treatment to get into the brain to effectively eradicate it in the brain? Do you know what I mean?
I do. You probably wouldn't give the treatment unless you know that the patient has involvement in the brain. Even if the subtype is more likely to go to the brain, it still might just be a minority of people that get that. So you wouldn't treat for a disease that a person doesn't have.
Got it. And a lot of that's going to be an observation with your patients, clearly speaking to them, you can tell, are they cognitively impaired? Are they experiencing, out of the blue symptoms neurologically, things like that. There's other factors that go into it, I think. So that if I were to go tomorrow to my doctor and say, which I tell everybody to do, and I want you to tell me how to improve and give better guidance to my friends, family members, and most importantly, everybody listening to this podcast and in our community, what should they be asking for? Because I tell everybody, go to the doctor, and you live in New York, you're hiking, you're in the woods, ask for a full tick-borne disease panel with an alpha-gal addition, because not everybody considers alpha-gal a tick-borne disease, right? Is that the proper way to ask your doctor? Because not everybody understands that. So first of all, is that the right way to ask for a broad test?
It's a good way, although it's complicated because doxycycline, as we were saying before, is a broad spectrum antibiotic. So I would be worried if someone came in, let's say they were hiking and they found a erythema migrans like rash. I'd rather that patient just get doxycycline right away. And we know that that doxycycline is going to take care of most things. It's going to take care of the Borrelia burgdorferi, the Anaplasma, the Ehrlichia, the Borrelia miyamotoi, the Borrelia mayonii. What it won't take care of is the parasite. So you could then recommend that they be tested for non-bacterial things, so Babesia microti, and it won't do anything for viruses. Unfortunately, though, the viral infections we don't have treatment for right now.
Right. The virals, you have to kind of help your body support that to put them
Yeah,
away, essentially, right?
exactly. It's supportive care. Mhm.
Supportive care. So I think you were describing a scenario where somebody's bit, they have a bullseye, they go to their doctor, get doxy, maybe ask for a panel for Babesia because Babesia is a parasitic bloodborne infection, but hit the treatment pretty quickly. Now, what about the scenario where you were active this summer? And I mean, this happened to 3 people in the last couple of weeks right here on Long Island. And Rich, and one of them was my closest friend's mother. She was active, was in the woods, started feeling really unwell. To me, it seemed very limey. Now I'm like, I always see Lyme because of what we do with Tick Boot Camp, right? But I'm like, just go get a full tick-borne disease panel.
Yeah.
Lo and
I
behold,
think that's
she's, she tests positive and she's getting treatment and she's feeling better. But is that the proper thing to ask your doctor for? Like in the scenario where you don't have a bullseye, but you think and you suspect because, where you live, or you just, you're feeling unwell, or you just want to do a routine annual checkup. What are the words you say to your doctor?
I see what you mean. I think that's a good recommendation. First of all, you need to tell your doctor, I've been exposed to ticks. I've been out and about, and I know that I'm in an area where there's a lot of tick-borne diseases, and I just wanted to let my doctor know, so I would just say, doctor, I have been exposed to tick bites. So that's a big thing right off the bat, because people don't always think that. But if you're really savvy, then yeah, you could say, well, you could ask questions. What is your procedure for testing for tick-borne diseases? Would it be possible to order a broad panel for tick-borne pathogens? Now, there's controversy about how broad you should go, because that could get really expensive. You could order all these tests, really the more targeted approach for the main things. That's probably all you need to do. So as I was saying, we have an algorithmic approach we recommend with a panel. So we'll test for Lyme disease, anaplasmosis, babesiosis. We don't usually test for Poisson virus right off the bat, although we have it as like a backup if patients, we like to do a tiered testing. So if the initial group of tests are not positive and the patient still has symptoms, then you can go on and order other things. But there's different ways of doing it. It all depends. We're trying to think of the patient here too. And you could spend a lot of money, thousands and thousands of dollars doing a lot of tests.
Right.
And so you have to really think of what's most likely to get me my answer for my patient. But I think the advice you're giving is good. Say, doc, I was exposed to ticks, and what's your typical way for testing for tick-borne diseases? And if your doctor says, no, I just do a Lyme disease serology, you could say, would you consider doing a full tick-borne panel for all the things that are endemic in this area?
Yeah, I think that, so I'll take it a step further. I've done this. I've gone to my doctor, and for me, if somebody has insurance and it's a copay for that blood draw and for all the tests, a flat copay, I say go to town. Whether it's 5 or whether it's 20, I don't care, right? It's a copay, so it depends on your personal situation as well.
Right,
whereas if you're paying for it, then you want to be maybe a little bit more cost sensitive, right? Mm-hmm. So I remember going to my doctor years ago and saying, I want the whole kit and caboodle. Takes his tablet, he goes, all right. And he showed me his tablet with all the tests, right? And he's like, check. And he's checking boxes on his tablet. I think this looks like a tick-borne disease, right? And he's just checking them off. But then what happens from there, right? So then I get the script, and what impacts where my blood goes and how it gets tested and what methodology is used at that point? Because does it matter if I go to a Quest versus a LabCorp? And then from there, does it matter what part of the country or the region it goes to? And then how does it get disseminated, right? And how do I know, like you said, one manufacturer versus another manufacturer? Like what happens behind the scenes after that?
Yeah. Well, it really depends on what laboratory your doctor's associated with. And if you are in New England, a lot of the labs there, a lot of the doctor's offices have agreements with laboratories that offer tick-borne disease testing. So if you're in, say, Boston, it might go to Massachusetts General Lab for testing, or Beth Israel, one of the Harvard systems. But if you are in more of a rural area, maybe it's going to go get sent off to a Quest or a LabCorp, and it's going to take a day to get there. It all depends on what's available and which laboratory your doctor has a relationship with. And it is true, not all tests are created equal. I would say all the big laboratories, though, have good quality tests. The question gets to be if it's a smaller lab that maybe isn't following all the best quality procedures. So it it's hard though, as you said, Matt. Well, as we were saying earlier, there are not FDA approved tests for a lot of these different things. So that means that every laboratory is creating their own tests, at least for the PCR tests. The serology tests are FDA approved, but the tests in my laboratory were developed by my lab, you know, under my
By you.
guidance. By me and my developer and my guidance. And I have confidence in my tests because I took the time to make sure they were really created well and really put them through the test to show that they were really good tests. But
Right, but if somebody goes to their
yeah,
doctor, is any doctor's office sending their blood to Dr. Pritt for a PCR test, or is it going to your colleague for an indirect test? What is that what's happening there?
yeah, it really depends. It depends on what algorithm your physician is going to order. If your physician, what tests your physician's going to order? Are they going to order just the Lyme serology, or are they going to order a whole bunch of different tests? And then it depends on which laboratory they send it to. So
So they have the
yeah,
flexibility to say I want PCR or I want antibody, the doctor.
the doctor could, yeah.
Got
Of
it.
course, it gets into all those problems with the PCR test for, say, Borrelia burgdorferi may not be as good as, it's not, if you're going to test from blood, it's not necessarily a good test or a good
Right.
specimen to test.
And when you said it matters where your doctor is partnered with, that's only if they're drawing the blood in the office. If they refer you to a LabCorp or a Quest, then it's
Hmm.
being the lab is Quest is the lab
Yeah.
or
And
LabCorp is
those
the
are good.
lab.
Right. And they're good.
Okay.
Those are good laboratories. They are, accredited by all of the high quality accrediting bodies like the College of American Pathologists. So you could trust in those.
Okay. So now let's finally get to the really, really, really fun piece, which is
Okay.
the metagenomics. I
Yeah.
mean, you got me so excited when you said that before. I'm like, I can't wait to get there. So let's talk about it. Can you re-describe what metagenomics testing is and where you're going and what you're doing with it?
Yeah, I mean, metagenomics, it's basically PCR on steroids. It's not even PCR. It's like our ability to detect all of the DNA and RNA out there. Um, Now, there's different ways to do metagenomics. You could sort of target it to a type of metagenomics that detects all bacteria or all fungi. But there's something called shotgun metagenomics. And I think it's because of the old fashioned shot, like a shotgun. It goes everywhere. It's like a big, like think of the Bugs Bunny cartoons where little shot goes out all over. That's why they call it shotgun metagenomics. It has the broadest coverage. Shotgun metagenomics, depending on how you set it up and do it, it has the potential of detecting any DNA or RNA that is present in that specimen, be it blood, synovial fluid, CSF, tissue sample, urine, whatever's there, it has the potential to detect. It will also detect human DNA. Now, we don't particularly want the human DNA in my lab. We usually try to get rid of the human DNA and focus in on the microbial DNA or RNA. Although in the future, looking at the human DNA might be interesting to see if the human host is maybe developing an immune response that would be indicative of Lyme disease. So
Fascinating.
yeah, it's really exciting stuff. We're doing it in my lab, but right now I think there's a few labs, not a ton, but a few labs in the United States that are using metagenomics tests, but they're not quite, we're all doing research to make them better. They're all kind of slow and expensive, and they're not necessarily good for Lyme disease. They're really good for some of the other things like anaplasma and Borrelia miyamotoi, but we're still trying to optimize them.
Okay. So from what I understand, metagenomics is a way to take a sample of blood and rather than segmenting and saying, do this and do this and do this and do this and rack up your bill, it's basically one path to do this one thing, which says look for everything. Instead of the
Right.
doctor saying, check for these 4 things for Matt, check for these 7 for Rich. It's just,
Right.
hey, go check for everything and let me know what comes back. Tell me what's going on with this guy, basically. Is that a good way to put it? Okay.
It is, yeah. Now,
So,
yeah.
go ahead.
Oh, no, go ahead.
No, I was going to say, so I think the hard part you're saying is it's looking at everything, including the natural part of our human body, right? The human genetics. So you have to basically filter out probably the majority of it, saying that's for now. You know, you talked about the benefit of that for maybe the future. Filter out the human side of it and say what's non-human, and then what is that? And that's the microbe, that's the virus, that's the bacteria, that's the parasite. And then you can just say Matt and Rich are both doing metagenomics. We both pop for 17 different things, but it's just all part of the same test versus a specialized doctor thinking Matt may have these 4, Rich may have these 7. It's more of a what the heck is going on here? I have no idea.
Right.
Okay.
And it gets a little bit more complicated because all around us are microbes. Like there's bacteria that live on my skin. There's bacteria that lives in the water. And so when you have a test that's that powerful and it casts such a broad net, it's going to pick up little fragments of DNA that might be in the equipment on the inside of my plastic tube that I'm using for testing. So the important thing with metagenomics is you have to be able to wade through all the noise to get to the pathogen. So if I find Borrelia burgdorferi in a patient's sample, that's a pathogen. But if I find Staphylococcus epidermidis, well, it's probably just skin flora. It's probably something I can ignore. Although in some people, it could be a pathogen. But you can see how it could get confusing.
Yeah.
So people are using artificial intelligence to scan through all of this. You need a really good bioinformatics system to help filter out the noise, find the true pathogens. And then as a physician myself and working with some excellent scientists and medical directors, we also can look at the patient's record and find out what's going on with that patient and help or use those results to help interpret what we're seeing. And then the exciting part I said, like for the future, is eventually we want to look at the human host response as well. There's some preliminary results that show that the human body produces RNA that we can detect when they have Lyme disease that's different than when they have babesiosis. So eventually we want to pull that in as well.
So you're saying the human side of it would be different to indicate an infection. So you're looking at the human side of the DNA metagenomics test to say, oh, that means Lyme, even though it's a human piece of DNA.
Yeah. It's the way the human's reacting and it's forming RNA to react in proteins. And you're basically looking at what the body's producing in response to whatever they're infected with. And that gives us information. Now, that work is very preliminary, but my lab is starting to look at that as well. But right now, what I'm most excited about with metagenomics is our ability to detect the known things, the things we already know about, but also the unknown things. And we can detect co-infections, we can detect viruses, not just bacteria. So it's a very powerful technology.
When you say unknown things, is this a tool that can identify other species that we have no idea even exist? Like you were saying earlier, when you kind of found these atypical things. So I'm going to use a simple an analogy, right? So I'm in the IT space, and I often say, man, these guys that are coming out of college have it so much easier with modern technology. I had to do it the hard way. And I feel like what you're telling me is, man, I had to find these new species the hard way. But with metagenomics in the future of science and medicine, they're going to get it the easy way. They're just going to have a tool to run it through the AI and say, oh, here's a new thing. Here's a new thing. You got this. It sounds like a similar analogy where we're getting better technology to result in better specific data to help people and to help people identify what's making them sick.
Yep, absolutely. The only difference I would say is that the PCR tests, the older tools, are usually a little cheaper and faster. Than compared to these metagenomic tools, which are a little slower and more expensive. But that's going to change. You know, any new technology starts off expensive. I mean, think about how much the first computers cost and how slow they were and what they did. You know, things are going to get faster, more powerful, and more affordable. It's just going to take a little bit of time.
When you said that the Lyme bacteria isn't great at being identified by metagenomics, why is that? Because that's something I found interesting. You said that earlier.
Oh, yeah, well, only because it's kind of like PCR. You're not detecting the DNA in blood in any large quantity.
Oh, because it's
So
not highly concentrated. Same reason.
Because
exactly.
of the sample.
Yeah,
Right.
Yeah.
it's the sample. Getting back to what we had talked about, Rich. It's not the test as much as it is the sample that just doesn't have a lot of organism in it.
Right.
Got it.
So, so now we have, I guess, a third type of test, right? So we have, we have our, or another type of direct test, right? Because I
Yes.
was breaking it down
Yep.
into direct and indirect, and we have the indirect test, which clearly I'm not a big fan of. I think I've made that clear.
Hmm.
We have our direct test, but we have another way of testing, and just so the way I'm
Yeah.
hearing the metagenetics is what you're doing with the PCR or the culture or the antigen testing is you're looking to say, can I find this thing? As
Hmm.
opposed to the metagenetic testing where you have the whole haystack and you're saying,
Yeah.
let's tease out the things that we don't want to look at. In this case, it's all the human DNA, right?
Hmm.
And now we have a profile of everything else there. But now the question is, how do you find the needles in the haystack, right? You're
Yeah.
not you're not just looking for the specific things. And then of course the other challenge that I think we're going to have with the metagenetic testing is, well, are we testing blood? And if we're testing blood, we're not likely to see a certain thing that we would want to know about. Are we testing urine?
Hmm.
And what are we going to see in urine versus blood? Or are we going to be testing some other fluids, or are we doing a testing tissue? I mean, what is it that we're testing? And there are going to be limitations to the quality of the result when we're looking for certain type of microbes if we're not testing the entire body.
Yeah, so you've hit on a lot of good points, Rich. So first of all, what specimen are we testing? And I think we're going to need to do some studies on that when we have this really powerful technology. There's also, are we trying to detect intact DNA or can we detect just small pieces of it and still make a diagnosis? And then are we going to detect only DNA? Or do we want to also look for RNA? Because you could do both with metagenomics. And are we going to try to get rid of all the human DNA first? There's some methods where you can do what's called host depletion. You try to get rid of all the human DNA so you only have the microbial DNA. So, how can we concentrate what we're looking for and get rid of the stuff that we don't care about? Of course, that's going to change if we start looking at the human host response and the transcriptome as well. And then there's things we could do, like we could add in probes, capture probes that will kind of go out and try to capture the things we want that aren't present in very high numbers, like maybe Borrelia burgdorferi. So there's a lot of things that we could do that my lab's looking at right now. And I'm working with my colleagues at Mayo. Dr. Ellie Thiel is brilliant. Dr. Patricia Simner, she's another colleague of mine. Dr. Andrew Norgan, and we're trying to ask all these questions right now to develop that test that will detect anything that's possibly causing harm to that patient in a single sample. That's the goal.
So you and Matt were talking about the AI element to the metagenetics tool. Can you
Yep.
build that out a little bit more for me? I wasn't clear on how the AI is assisting you in, I guess, finding the needles in the haystacks.
The AI tool is really for the bioinformatics because a metagenomics tool produces so much information, including all the contaminating DNA, the stuff that was in the water, the reagents, the plastics. So the bioinformatics can use artificial intelligence to help sort out the things that are junk versus the things that are real. Because sometimes the things that are real are present in very small numbers, and you have to really do a deep sequence to try to find them. But then that means you have so much data that it's kind of like a human can't really, in any small amount of time, sort through it all. You need that bioinformatics to help you sort through the vast amount of data to find the stuff that's really important.
So, for example, you would know what type of a tool you were using to take the sample, and then you would know to tease out the plastic, for example, of a particular tool you were using to take out the sample as
Mhm.
part of the AI analysis. So you're teasing out the human DNA, you're teasing out the particular type of plastic that was used, or
Mhm.
and you just
Yes.
keep taking out all of these.
Subtraction. Right.
Wow,
And
that is so cool.
And what we'll do, one of the ways we do that is we run a negative control with every specimen, of course, and you expect it. Well, normally a negative control would be truly negative, but because we're detecting little bits of all this DNA and RNA, even in, like I said, the reagents, the saline, the water, the various components we're using, we would expect the negative control is going to pick those up. Okay, so if the negative control picked up these 5 things, but we know it's negative because we're not testing any human sample, then we know that those 5 things are contaminants. So we can subtract them from the patient samples as well. So there's some tricks like that that we could do.
Hmm.
And there's some
Wow.
environmental stuff. There's some bacteria that live in the soil that we know don't cause human disease. And so some of those we could subtract out easily as well.
Dr. Pritt, real quick, when you said the probe earlier, so you said Lyme is hard to find, and we have tools like doing a probe to go
Yes.
find the Lyme using metagenetics. What does that mean? Can you describe that a little bit more?
Yeah, it's a capture probe. It's one way you could basically, it's a probe that has a piece of DNA. It's a DNA probe, and it will be a perfect match to, let's say, a part of Borrelia burgdorferi. And by capturing it and then amplifying it, it kind of just increases the signal, so we're more likely to detect it then.
Hmm. That's
Yeah.
fascinating. The
That
one
is
thing I
that.
forgot to ask is we said this multiple times. Can you just describe the difference between DNA and RNA?
Yeah, DNA is the backbone of what our body and mammals and animals uses, eukaryotic cells. It's really viruses that will use RNA as their primary genetic code. But humans, we use DNA. It's double-stranded. The DNA is our library, our code. It tells the body what we're going to make. So the DNA is used to tell our body using RNA how to make different proteins, for example. So they're
That,
both, yeah, DNA and RNA. So when I'm talking about detecting RNA, I'm either talking about what the body's using to make proteins, or I'm talking about RNA viruses, which we know are a problem. Powassan virus is an RNA virus, Heartland virus, Bourbon virus. So we want to detect those as well.
so we were excited to interview you. We had very high expectations. You've exceeded our expectations. And as much as I would love to keep you for another 2 hours, it wouldn't be fair to you on a Friday night to spend more time with us. So we would like to invite you back at some point in the future so that we can have a follow-up, because there are many, many questions that we weren't able to get to in this 2 hour window, but you've been very, very kind to share so much of your time with us and with our listeners and our viewers. So thank you for all the great work you're doing. Please stay in touch with us and let us know how everything is developing with this metagenetics, because I'm very, very excited about that approach. And I know folks who are listening to this are certainly very excited that this could be the breakthrough that we're all looking for improving the horrific testing methods that currently exist in this community. Again, thank you for all the great work that you're doing.
Well, thank you, Rich. Thank you, Matt. It's been a pleasure. You guys are great. You are really up on things, and it's been a pleasure talking with you. Hope you have a great night and a great weekend, and I will be definitely keeping in touch.